Plant hormones. How do roots know to grow down, and how do stems know to grow up? How do the tissues of a plant determine the right time of year to produce flowers? In short, how do the collection of cells in a plant manage to act together as a single organism? Is something carrying messages from cell to cell? Hormones are chemical signals produced by living organisms that affect the growth, activity, and development of cells and tissues within them. They also coordinate responses to the environment. In plants, hormones may act in the same cells in which they are made, or they may travel to different cells and tissues as seen in the figure. So the hormone producing cells are in the tip, and the movement of the hormone comes down and triggers cells in another bud lower down, a secondary bud lower down. One mechanism of hormone action in plants is shown. In this case, hormone producing cells in a mature flower release hormones that travel into the flower buds and inhibit development of the buds lower down. Once the mature flower is done blooming, production of the inhibiting hormone will decline, and the flower bud below can then begin its bloom. A portion of an organism affected by a particular hormone is known as its target cell. To respond to a hormone, a cell must contain hormone receptors, usually proteins, to which hormone molecules bind. The response that results will depend on what kind of cell receptors are present in the target cell. Depending on the receptors present, a given hormone may affect roots differently than the way it affects stems and flowers, and the effects may change as the developing organs add or remove receptors. Charles Darwin and his son Francis explored the mechanisms behind the grass seedling tendency to bend toward light. The results of their experiments suggested that the tip of the seed somehow senses light. The Darwins hypothesized that the tip produces a substance that regulates cell growth. So this is the control group, and the sun is over to the right. With the tip removed, there was no bending. With a tip covered in an opaque cap, there was no bending. With a clear cap, there is still bending. And with an opaque shield over the base did not bend. The hormone auxin stimulates cell elongation and the growth of new roots, among other roles that they play. They are produced in the shoot apical meristem and transport to the rest of the plant. Where light hits one side of the shoot, auxins collect in the shaded part of the shoot. This change in concentration stimulates the cells on the dark side to lengthen, causing the shoots to bend away from the shaded side and toward the light. More auxin, it will extend the cells, and less auxin will not. Auxins also regulate cell division and meristems. Growth at lateral buds is inhibited by auxins. Because auxins move out from the apical meristem, the closer the bud is to the stem's tip, the more it is inhibited. Lateral buds near the apex grow more slowly than those near the base of the plant. This phenomenon is called apical dominance. So here's the shoot tip at the top, the dominant bud. So these auxins are released, they're concentrated in the shoot, and they flow down the stem. Here's an auxiliary bud, which is inhibited by these auxins. But if that apical meristem is removed, the inhibiting auxins are removed, then this bud is triggered. And so a new shoot on the auxiliary bud location grows. So again, if you snip off the tip of the plant, the lateral bud begins to grow more quickly and the plant becomes bushier. This is because the apical meristem, the source of the growth inhibiting auxins, has been eliminated. Cytokinins flow up from the roots, but they are suppressed by the high concentrations of auxins. This causes buds below not to grow. So the cytokinins are blocked by the auxins. But when it's removed, the cytokinins are triggered to activate a lower bud since auxin concentration is reduced. So when auxins are then produced in this new bud, the expanding shoot tip, then their high concentration of auxins suppress the cytokinins once again, so that buds even lower will not develop. Cytokinins are plant hormones that are produced in growing roots and in developing fruits and seeds. Cytokinins stimulate cell division and they interact with auxins to control the growth of new organs in injured plants and to balance root and shoot growth. Cytokinins also delay the aging of leaves and play important roles in the early stages of plant growth. Cytokinins often produce effects opposite to those of auxins. Auxins stimulate the initiation of new roots and they inhibit initiation and growth of new shoot tips. Cytokinins do just the opposite. If a tree is cut down, the stump will often make new shoots because cytokinins accumulate near the cut while auxins move away. Hormones called gibberellins stimulate growth and can influence various development processes. Gibberellins promote germination of plants and may cause dramatic increases in size, particularly in stems and fruits. Gibberellins are found in the meristems of shoots, roots, and seed embryos. Gibberellins interact with another hormone, abscisic acid, that controls seed to dormancy. Abscisic acid inhibits the cell division, thereby halting growth. Abscisic acids are in the terminal buds and seeds. When the seed development is complete, abscisic acid stops the seed's growth and shifts the embryo into a dormant state. The embryo rests until the environmental events shift the balance of hormones. Some events may include a strong spring rain that washes abscisic acid away. Without the opposing effect of abscisic acid, the gibberellins can signal germination. Abscisic acid and gibberellins have opposite effects, much like auxins and cytokinins. In response to auxins, fruit tissues release small amounts of hormone, ethylene, which stimulates fruits to ripen. Ethylene also causes plants to seal off and drop organs that are no longer needed. For example, petals after flowers have been pollinated, leaves in autumn, and ripened fruit. Ethylene signals cells at the base of the structure to seal off from the rest of the plant by depositing waterproof material in their walls. What are some examples of environmental stimuli to which plants respond? Plants have sensors that detect the direction of stimulus and signal elongating organs to orient their growth toward or away from these stimuli, such as light, gravity, and touch. These growth responses are called tropisms. The tendency of a plant to grow toward a light source is called phototropism. Changes in the concentration of auxins are responsible for phototropism. Experiments have shown that auxins migrate toward shaded tissue, possibly due to changes in membrane permeability in response to light. Auxins also affect gravitropism, the response of a plant to gravity. Auxins migrate to the lower sides of the horizontal roots and stems. In horizontal stems, the migration causes the stems to bend upright. In horizontal roots, however, the migration causes roots to bend downward. Some plants respond to touch, a process called thigmotropism. Vines and climbing plants exhibit thigmotropism when they encounter an object and wrap around it. Other plants, such as grapevines, have extra growth called tendrils that emerge near the base of a leaf and wrap tightly around any object they encounter. Some plant responses are very rapid. A mimosa plant will respond to being touched by folding in its leaves quickly. This response is produced by decreased osmotic pressure in cells near the base of each leaflet. The carnivorous Venus flytrap also demonstrates rapid response. When an insect lands on a flytrap's leaf, it triggers sensor cells on the inside of the leaf, sending electrical signals from cell to cell. A combination of changes in osmotic pressure and cell wall expansion cause the leaf to snap shut, trapping the insect inside. Photoperiod in flowering. As seasons change, most plants respond to a relative length of light and darkness, a stimulus called photoperiod. Photoperiod is a major factor in timing of seasonal activities such as flowering and growth, as shown in the figure. A plant pigment called phytochrome is responsible for photoperiodism and regulates the changes in activity that prepare many plants for dormancy as winter approaches. Phytochrome absorbs red light and activates a number of signaling pathways. So the effect of photoperiod on flowering, a short day plant affected by a long day, no effect. A long day plant, flower when exposed to a short period of darkness. For short day plants, they respond to a short day with plant flowering only when exposed to extended periods of darkness. No effect on a long day plant. An interrupted night, no effect on a short day plant, but a long day plant also flower if the brief period of light interrupts a darkness. This essentially divides one long day into two short nights. How do plants respond to seasonal changes? As cold weather approaches, deciduous plants turn off photosynthetic pathways, transport materials from leaves to roots, and seal off leaves from the rest of the plant. Many flowering plants lose their leaves during the colder months. The phytochrome in leaves absorbs less light as days shorten and nights become longer. Auxin production drops, but the production of ethylene increases. As chlorophyll breaks down, other pigments, including yellow and orange carotenoids, become visible for the first time. The brilliant reds come from anthocyanin pigments that are freshly made. Hormones produce important changes in apical meristems. Meristems produce thick, waxy scales that form a protective layer around new leaf buds. Enclosed in its coat of scales, a terminal bud can survive cold winter days. At the onset of winter, xylem and phloem tissues pump themselves full of ions and organic compounds. The resultant solution acts like an antifreeze in a car, preventing the tree's sap from freezing.